In this lab exercise, we will be performing the polymerase chain reaction. So the polymerase chain reaction, otherwise known as PCR, is actually an in vitro technique for cloning of DNA. So when we say we are cloning a DNA fragment, we are actually making several copies of the DNA. Early on, the only way to copy DNA was to insert this DNA in bacteria and allow the bacteria to replicate. And with each cell division, your DNA is copied. So in 1983, Kary Mullis, a biochemist, had thought of an idea of using a pair of primers to bracket the desired DNA sequence and copy it using DNA polymerase. So this technique would allow a small single strand of DNA to be copied almost an infinite number of times. For this, he won the Nobel Prize in Chemistry just 10 years after his invention in 1993. So the initial reaction mixture includes a very small amount of double-stranded DNA to act as a template. You have your deoxyribonucleotides or your DNTPs. So this is deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxythymidine triphosphate, and deoxycytidine triphosphate, which are the monomeric components of your DNA. Very important are the oligonucleotide primers, which will flank the region or bracket the region of DNA to be amplified. So they are complementary sequences to the ends of the DNA you want to copy. The polymerase which was used here is derived from a thermophilic bacterium, Thermus aquaticus. So it is called your Taq DNA polymerase. What is significant about your Taq DNA polymerase is that it is heat resistant. Because the organism from which it was derived lives in high temperatures in hot springs, then the enzyme is very active at high temperatures and is not easily denatured. And all of these components are mixed together in specific buffered conditions. So PCR is also described as a thermal cycling technique. If you take a look here, these are the steps in PCR. So you start out with your template. Increasing temperature denatures your template, exposing the single strands to which your oligonucleotide primers can bind to, and then increasing the temperature to the optimum working conditions for the Taq polymerase allows the polymerase to extend the primers, leaving you with two new strands of double-stranded DNA. So if we repeat or cycle these temperatures, denaturing, annealing, elongating, then after 20 to 40 cycles, you will have almost millions and millions of copies of DNA to end up with. That is why it is called a thermal cycling technique. So it's thermal cycling because PCR uses repeated rounds of denaturation, annealing, and extension to amplify DNA. PCR is carried out in a PCR machine, which essentially is a heating block capable of cycling those temperatures very quickly. So if you take a look at the thermal profile of a single PCR reaction, you have again very high temperatures for denaturation, lowering the temperature to allow hybridization of the primers through their complementary sequences, and again, an increase in temperature to allow your DNA polymerase to elongate the primers. So each cycle of the PCR doubles the amount of DNA in the previous cycle. So it is actually an exponential increase in copies of the DNA. Theoretically, if you start out with 1, after 1 cycle you have 2, after 2 cycles you have 4, after 3 cycles you have 8. So after n cycles, you have 2 raised to the n number of DNA copies if you had started with a single copy of the template. So you will see that because of the use of a pair of primers bracketing the ends of the DNA you want to amplify, after so many cycles, the predominant DNA in the reaction is identical to the sequence which is specified by the primer pair. So what are the components of your PCR? Very briefly, the PCR mix is composed of your DNA template, a pair of primers, your DNA polymerase, your nucleotides, and the buffer, which may contain the cofactors for the activity of your DNA polymerase. So there are several considerations when determining the concentration of each of these components in your PCR reaction. For example, for your DNA template, how much template is needed? Theoretically, and what actually makes PCR a very revolutionary technique is that you only need very little of the DNA template to begin with. It's not necessarily the more the better in this case, because a high concentration of DNA template in the PCR reaction can actually lead to non-specific amplification. So because there's too much DNA, your primers and your DNA polymerase might not be so specific, and the polymerase might be very active, such that it leads to the amplification of non-target template. You have your DNA polymerase, and as I said initially, the most common and the first polymerase which is used for PCR was derived from Thermus aquaticus. However, now there are several different polymerases also used in different PCR reactions. Some of them have higher fidelity or are more accurate than your Taq DNA polymerase. Some of them are more active, have a higher activity than your Taq DNA polymerase. So the choice of polymerase is often dependent on the end use of your PCR product. You also have your oligonucleotide primer pair. So it's a pair of primers referred to as the forward and the reverse, each specific or complementary to the 5' and the 3' end of the sequence that you want to amplify. Primers are designed with prior knowledge of the sequence that you want to amplify, and primer design is very important in the success of your PCR. So you also have your deoxynucleoside triphosphates. So as I mentioned, those are DATP, DGTP, DTTP, and DCTP, which are the nucleotide monomeric components used by the DNA polymerase to synthesize the new strand of DNA. So all of these are placed in a specific buffered condition at a specific pH, which allows the optimum uh, working conditions for your DNA polymerase and the stability of the DNA template and the amplification product. Often, part of the buffer are cofactors, ion cofactors. More specifically, magnesium ions are provided in the buffer because magnesium is a cofactor of DNA polymerase. The amount of magnesium can also dictate the specificity of your DNA polymerase. Again, there is an optimum concentration of this cofactor which is required for the optimum activity of the DNA polymerase. Too high magnesium ion concentration can cause, again, non-specific amplification. However, having too low magnesium ion concentrations can prevent the DNA polymerase from working properly. So PCR experiments often have to be optimized so that very stringent conditions will yield specific products. So optimization can start with determining the optimal concentrations of your PCR components. And this is very important so that your PCR yields the target product, target amplification product, and does not lead to non-specific amplification or the amplifications of non-targets.